Hey everybody, and welcome to week 2 of our AMP1 online lecture series. Today's material will be covering chapter 23, unit 3 through 5, or module 3 through 5 in chapter 23. And we're going to be continuing to talk about metabolism. So again, just a brief overview of what we're talking about is either anabolic processes that synthesize something larger from a smaller precursor. And an important part of anabolism is, of course, that it requires energy input to make this reaction occur. And again, the synthesis and an anabolic process are the same thing. And the opposite of that being catabolism, or breakdown, where instead of forming new bonds, you are breaking bonds. And releasing potential energy that can be stored in the form of ATP. So these are our two our two reactions that we find in metabolic processes, and we're going to finish up talking about catabolism by looking at what happens when you have things like amino acids and fatty acids, and how are they catabolized. We've talked to a large extent so far about glucose and how it's metabolized, or how it's broken down, and how that feeds the electron transport chain producing ATP. We'll talk about how amino acids and fatty acids can do the same thing, and we'll also start to get into and actually complete a discussion about anabolic processes. What are the synthesis reactions that are taking place in the body? So in getting into finishing up our catabolic processes and talking about how fatty acids are catabolized, how do we get energy out of fats? Well, the stored form of a fat, or the form that we are usually ingesting, is going to be this molecule, the triglyceride. And what you may remember from talking about macromolecules is that a triglyceride is really built from two different components. You have a molecule of glycerol, and you can see glycerol, this molecule has 3 OH groups. You have that molecule of glycerol that's joined with 3 fatty acids, and those fatty acids, remember, have these long hydrocarbon tails. And those hydrocarbon tails are actually where the majority of the energy is going to be produced, is by breaking the bonds between the carbons in those tails. But these two molecules, glycerol and the fatty acids, are joined together to make the triglyceride. One glycerol molecule joined to 3 fatty acids. So when you have a triglyceride, and the metabolic needs of the body are that there's an energy need, the triglyceride that's stored in your adipose tissue, or that you ingest in your diet, is going to be broken down into these components. And that process is called lipolysis. So that term lysis, again, just means breaking down, and then lipo just refers to the fact that this is a fat, this is a lipid that we're talking about. So lipolysis is breaking down a triglyceride into glycerol and fatty acids. Now, both of these can actually be used in further catabolic reactions to generate energy ultimately. So now moving on and talking about what happens to those molecules that we've just produced after lipolysis. So you have some energy need, you need to break down your triglycerides, and you go through the process again of lipolysis, breaking down one triglyceride into one molecule of glycerol and 3 fatty acids. So what happens to these? What happens with glycerol is actually quite simple. There's a conversion of glycerol to the molecule G3P, which hopefully you remember from our last lecture stands for glyceraldehyde 3-phosphate. So this is a 3-carbon molecule that results from breaking down glucose. And glucose is broken down by addition of phosphate groups that requires ATP input. That's the energy investment phase in glycolysis. So G3P is what's left over after you split glucose when you're getting to the energy payoff phase. So G3P is going to be able to enter, it's going to bypass the energy investment phase of glycolysis and go straight into the payoff. So glycerol can be used directly to generate ATP through glycolysis without any extra ATP being used to get to this point, to accumulate glyceraldehyde 3-phosphate. And so this is going to provide about 5% of the total energy that you'll produce through catabolism of a triglyceride. So after the triglyceride has been broken down into glycerol and fatty acids, now let's talk about what happens to the fatty acids. What's going to happen is a process called beta oxidation. Now remember, oxidation again is transfer of electrons to another substance. So oxidation of fatty acids, which have lots of electrons to give up, is going to result in the formation of NADH and FADH2. Now again, those are the reduced coenzymes that are carrying electrons to the electron transport chain. And that's what happens to provide the bulk of energy that we get from breaking down sugar. So same with fatty acids. The point in breaking them down is ultimately to generate as much of these two as possible. In addition, you're also going to create acetyl-CoA, which hopefully you remember can enter the Krebs cycle to generate even more of these two substances. So we're really using beta oxidation to generate lots of these electron carriers that can then take those electrons to the electron transport chain. And this is going to drive the formation of about 95% of the ATP that we get out of a triglyceride. It's going to be from beta oxidation of those fatty acid tails. So one alternative that can happen, and you may, if you're up to date on nutritional information, you may have heard about ketogenic diets, which are low calorie or low carbohydrate intake diets. So what ketogenesis is the formation of ketone bodies. So this acetyl-CoA that builds up under certain conditions, if you're starving, for example, or you take in extremely limited amounts of calories or carbohydrates, your fats are going to be broken down through beta oxidation to produce acetyl-CoA. Now, acetyl-CoA can't be transported efficiently in the blood, but what can be is a ketone body. So acetyl-CoA in the liver will be converted to ketone bodies through this process of ketogenesis. Now, ketone bodies in the blood are going to go out to all the tissues that need to produce this energy, again, under the circumstances where you're starving or whether you're severely restricting calories or carbohydrates. So you're making lots of these ketone bodies, and what happens is once they arrive at the tissue, they're going to be converted back into acetyl-CoA that can enter the Krebs cycle and generate these electron carriers. Now, if this goes on for too long, in addition to acetyl-CoA entering the Krebs cycle, there are also other substances that need to enter the Krebs cycle along with acetyl-CoA. And those substances tend to come from carbohydrate metabolism. So if you're starving or on a severe calorie or carb restriction, you're going to run out of those other components of the Krebs cycle, aside from acetyl-CoA. So what will happen is eventually acetyl-CoA is going to build up enough so that it'll actually stop the conversion of ketone bodies into acetyl-CoA because it's not going to be able to proceed through the Krebs cycle. So again, this is in very extreme cases of starvation where you'll actually get a buildup of ketone bodies. And ketone bodies have an acidic nature, and if they build up in great enough amounts, they can lower the pH of your blood to potentially dangerous levels. And we call this ketoacidosis. So in that case, your body will be trying to get energy by metabolizing fats, producing acetyl-CoA through the use of ketone bodies. And ultimately, that's, again, to produce as many of these electron carriers as possible. But in the extreme conditions, those ketone bodies can build up and actually reduce the pH to dangerous levels. So these are the catabolic fates of fatty acids and triglycerides in general. Next, I'd like to talk about how amino acids are catabolized. So you know a lot about the synthesis of proteins from amino acid monomers, but you may also find it interesting that we can use amino acids themselves to produce energy in the case that we need to. So what an amino acid is, once you've broken a protein down into its component amino acids, you can look at the basic structure of an amino acid as being some carbon skeleton, which I have represented here with this circle AA for amino acid. But the amino part of the term amino acid comes from the fact that amino acids have a amino group, or one nitrogen with two hydrogens. That's called an amino functional group. And this amino group has to be removed from the amino acid before it can be oxidized. So nitrogen is actually a very inert element. It does not react. And chemical reactions are required in order to oxidize something, to break bonds. So in order to make this carbon skeleton reactive, you have to first remove this amino group. And we'll talk about that more in just a second. So once you remove this amino group, what you're left with is the carbon skeleton that has lots of electrons that can be ripped off, and it has components that can be used for other purposes. So those carbon skeletons can be converted to a number of different useful molecules. For instance, pyruvate is a molecule that you can make from some of these carbon skeletons. And remember, pyruvate is what's going to enter the mitochondria in oxidative phosphorylation or cellular respiration to make ATP. You can also make acetyl-CoA out of the carbon skeletons of amino acids, and acetyl-CoA can just enter right into the Krebs cycle to produce lots of NADH and FADH2. Other Krebs cycle intermediates that we just mentioned can be made from glucose. You can make Krebs cycle intermediates also from the carbon skeletons of amino acids. And again, the Krebs cycle is going to produce lots of NADH and FADH2. And then finally, some amino acids are able to be converted directly into new glucose. So they can be catabolized into a form where new glucose can be produced from those amino acids. And we would call an amino acid that can be turned into glucose, we would call that a glycogenic amino acid, or excuse me, a glucogenic amino acid. So those would be the fates of the carbon skeleton, and that's very useful. So the process that removes that inert nitrogen, the amino group part of the amino acid, is actually going to be a process called transamination. So trans means that amino acid is not necessarily being removed. That would be a process, if we're simply to remove it and cast it off into space, that would be called deamination. This is transamination, and what it's doing is transferring this amino group to a different molecule called alpha-ketoglutarate. So when you aminate, when you add an amino group to alpha-ketoglutarate, what you get is a molecule called glutamate that contains now that amino group. So it's taken it from that amino acid. Once that occurs, you get deamination of glutamate. So where we remove now that nitrogen containing group, that amino group, from glutamate. And that's going to be a process called oxidative deamination, is removing that amino group, that now an NH3. So when you deaminate glutamate, you're going to reform alpha-ketoglutarate, and you're also going to release a molecule of ammonia, which is NH3. So the body processes ammonia into urea, which is then expelled in the urine. So if enough urea builds up, it's toxic to the body, so we need to get rid of it. And so amino acid catabolism is actually what's going to be forming urea, which again is clearly excreted in the urine. So now that we've wrapped up looking at our catabolic processes that involve the breakdown, let's talk about synthesis reactions, also called your anabolic pathways. So there are three primary reasons we would use an anabolic reaction. One would be to store nutrients. So if you have an excess of glucose, what are you going to do with it? You're going to store it for later use. And you can do that by forming things like glycogen. You can store energy in the form of triglycerides. In the case that there's an energy requirement, you can also synthesize new nutrients. So for example, gluconeogenesis is the formation of new glucose. So if there is low blood sugar, you can boost blood sugar, for example, by making a new nutrient, by making glucose from scratch. And you can also think about the synthesis of structural elements in the body, your proteins, your phospholipids that make up the cell membranes, and also your nucleic acids would be considered a structural element that is formed through a synthesis or an anabolic reaction. Now let's talk about specifically the anabolic processes that relate to glucose. So if there's an energy excess and we need to shift away from using glucose to storing it, we're going to go through the process of glycogenesis. So that is just the formation of glycogen, which is taking these glucose monomers and stitching them together, connecting them into this branched structure called glycogen. So this is going to be taking place in the liver and in skeletal muscle, are the two cell types that are going to be making glycogen. And glycogen is a really short-term energy storage molecule. It's not going to last very long if there's a high energy demand. So let's say you're really vigorously exercising and you've got no food on your stomach. Your muscles are going to use up your glycogen stores pretty quickly. And after that, they'll have to find other means of raising your blood sugar or producing glucose to use for other catabolic reactions. So the process of breaking down glycogen into sugars that can then be used for energy is called glycogenolysis. The other way that you can come up with glucose is through making glucose from scratch, gluconeogenesis. That means the new formation of glucose. And this happens in both the liver and the kidney. And what's going to go into these reactions, making new glucose, is going to be molecules like pyruvate, lactic acid, some of those glucogenic amino acids that I mentioned previously. Some amino acids can be converted to glucose. And some of the materials that you'll find as intermediates in the Krebs cycle can also be used to generate new glucose. So we can break down glycogen, or we can make new glucose. Those are the ways that we can raise our blood sugar through glucose anabolic methods. And another important thing to note here in gluconeogenesis is glucose is a 6-carbon molecule. These molecules that are being fed into this reaction are going to be 3 or 4 carbons usually. So pyruvate, lactic acid, for example. Those are 3-carbon molecules, so it takes a couple of those to make one glucose molecule. The anabolic processes that are used to generate triglycerides to store energy in the form of fat, of course relates to the fatty acid anabolic processes. This is called lipogenesis, and it's going to take place in the cytosol, places like adipocytes, or adipocytes, which are the fat storing cells. So basically, in fatty acid anabolism, you can take most any nutrient, so products of glycolysis, for example, so that sugar is one nutrient that can feed the generation of a triglyceride. Most any nutrient can be converted to acetyl-CoA and some of these other products of glycolysis. Acetyl-CoA molecules, in this case we're talking about those that come from glycolysis, but acetyl-CoA can come from a lot of different places. Acetyl-CoA can be turned into fatty acids, can be modified and stitched together to form those fatty acids, those long chains. And other products of glycolysis can be used to make glucose, which is in turn going to be turned into glycerol. And a fatty acid is going to be added to that glycerol backbone, 3 of them, to make a triglyceride. So this is how fatty acids come to be, triglycerides come to be, is unused nutrients being converted into a good storage molecule, which triglyceride is a very good one. Amino acids in the body, we know that one form of amino acid catabolism is protein production. But actually, going back to the synthesis of new nutrients, like you saw with glucose with gluconeogenesis, you can make new amino acids from scratch as well if you need them, if your body needs them. So of the 20 total amino acids, only 9 of those are what we would call essential amino acids, where it's essential that you get them from your diet. The other 11 of those 20 amino acids, our bodies have the ability to synthesize. So we don't necessarily need those 11 amino acids directly from our diet because we can produce them on our own anabolically. So when there's an excess of amino acids are not stored in the same way that glucose can be stored as glycogen or that triglycerides store energy. Amino acids can be converted to proteins, and if our body has met its protein requirement, if we've got more than enough protein, those amino acids can be converted through anabolic processes into glucose or fatty acids. And those are 2 molecules that can definitely be stored if there's an excess. So it's at this point that I would like to direct you to a really excellent image in your book that's going to show the big picture of nutrient catabolism. So what do we do with the nutrients? How do we produce new nutrients? On page 923 in your book, you'll find the figure called the big picture of nutrient anabolism. And I think that this is probably the best synthesis, no pun intended, of this information is up to this point, speaking about anabolic reactions. It's in this figure on page 923. Now that you're familiar with the different sets of reactions of metabolism, I want to talk about the different states where your body can shift between anabolic and catabolic reactions and why that is. And that it relates to how long it's been since your last meal. What your body is doing, metabolically speaking, relates to directly when you last had that last meal. And the two metabolic states that your body oscillates between are going to be called the absorptive state, which takes place directly after a meal and for several hours after that meal, and the post-absorptive state, which is going to take place after about 4 hours post-meal. In the absorptive state that takes place directly upon eating a meal and proceeds for several hours, you're going to get boosts in levels of insulin. And this is because as you digest that meal, if that meal contains carbohydrates, your blood sugar levels are going to rise. And lowering that blood sugar level is a product of the effect of insulin, which again is the hormone to control blood sugar levels, the antagonist of glucagon, which we'll mention later. So what's happening in the absorptive state is those nutrients that you take in from your meal are going to be oxidized to produce ATP or fuel the cells. But you've also got an excess of protein, glucose, and lipids probably from this meal. So you're going to be doing things like protein synthesis to build up the structure of your cells. You're going to be going through glycogenesis, taking excess glucose and making glycogen out of it, and using other excess nutrients to produce new lipids through lipogenesis, or to make new triglycerides for energy storage. So these are going to be mostly your synthesis reactions that are favored during the absorptive state. So in the post-absorptive state, when it's been a while since that last meal and you need to start using stored nutrients, you're going to be favoring the more catabolic reactions, so breaking things down. And the post-absorptive state, like the absorptive state, is going to be controlled primarily by hormones. So three main classes of hormones. You have cortisol, glucagon, and norepinephrine and epinephrine. So cortisol is going to favor the production of new glucose. It's going to stimulate gluconeogenesis. Glucagon, you already know, is used in opposing effect, the effect of insulin, to raise blood sugar levels. And it does this by stimulating the liver to, again, go through gluconeogenesis, but also to release that stored glucose that the liver has in abundance in the form of glycogen. So kind of the short-term answer would be to break down that glycogen, and the long-term answer to have raising blood sugar levels is through gluconeogenesis, making new glucose from scratch. And then norepinephrine and epinephrine are going to stimulate muscle glycogenolysis to break down glycogen and release stored glucose. But also it's going to stimulate lipolysis, primarily in adipose tissue. So lipolysis, again, is breaking down your triglycerides and proceeding with catabolic reactions to liberate energy from those fatty acids primarily. So just some general features of the post-absorptive state. You're going to see things happen like slowing down generally of anabolic processes. So you're not going to be building too many things during the post-absorptive state when you're at a loss for nutrients. You're also going to have things like proteins being broken down, and not necessarily so that they can be catabolized directly for energy, but indirectly because some of those amino acids in those proteins are glucogenic. They can be used to make new glucose, which can then be catabolized to make ATP. So proteins do provide a potential source of energy through release of those glucogenic amino acids into the blood. You're also going to get ketogenesis, which again is an alternative energy source where acetyl-CoA is converted into ketone bodies by the liver. And many cells in your body can use ketone bodies for fuel. And we'll get to that in just a second. you're also going to have to raise those blood sugar levels. As we just mentioned in the last slide, you have gluconeogenesis and glycogenolysis, both raising blood sugar levels. You have lipolysis, releasing fatty acids and glycerol from triglycerides. And all these nutrients are going to end up being oxidized to produce ATP. So the one thing about ketone bodies, they can be used for fuel in many cells in the body. However, the nervous system, which is primarily composed of neurons, really doesn't like to use ketone bodies as a source of energy. So you get this concept called glucose sparing, where cells stop using as much glucose as they normally would, so that it can be diverted to the neurons that truly need that glucose. So your brain really likes glucose and really doesn't like ketone bodies. So your body will use glucose sparing to preserve glucose for your neurons in the case that it's been a while since a meal. So now I'd like to talk about the regulation of feeding. What is controlling your mind to tell you whether or not you're hungry, whether you need a meal or not? So it's actually going to be based on nervous impulses from your brain, specifically in the hypothalamus. So within the hypothalamus, there are two what we call nuclei, which are the centers of your hypothalamus, clusters of neurons that are functionally related to one another. So there are two nuclei. There is the satiety center, which is going to control a feeling of fullness, and you have the hunger center, which is going to make you feel hungry. And these two centers are going to be considered either anorexigenic. So you may have heard of anorexia. That means you're not going to be eating very much. The satiety center is going to make you feel satisfied. It's going to make you feel that you don't need a meal. Whereas the hunger center would be orexigenic. It makes you feel that hunger. It's going to reinforce the fact that you need a meal soon. And these are both going to be controlled ultimately by hormones. So they will stimulate different responses physiologically to make you feel hungry. But those centers in your brain are going to be controlled by the hormones leptin, which is produced by adipocytes, or ghrelin, which is produced in your stomach. So what leptin will do to your satiety center is it's going to inhibit the hunger center neurons while also stimulating the satiety center neurons. And this combination of effects of leptin are going to make it so that you are feeling satisfied, you are not hungry. And this will be what's occurring after a meal, directly after a meal. Whereas the hunger center, which is stimulated by the hormone ghrelin produced in your stomach, is going to, when those hunger center neurons are active, is going to make you feel hungry. So you're more likely to go out and seek out that meal that your body has deemed is biologically necessary. So that'll conclude this week's discussion. I'll be posting some homework. Already posted some dynamic study modules for you to accomplish. So hopefully you'll be able to use those to help you work through this material. And I'll also be giving you any updates through email as I normally do.